Hello students, this is Dr. Faizan Mirza. In this video, I'll be discussing questions related to nucleic acids and protein synthesis. So I'm taking this question from Major in 2018, variant 22. They had these are the Watson and Crick, and the Watson and Crick were the scientists who got Nobel for coming out with the explaining how the DNA actually looks like into a spiral double helical structure having complementary base pairing. They say that Watson Crick used results to work back at about another scientist, and he found that the proportion of base ATGC were different in different species, but within the species, the proportion of A was equal to proportion of T, and proportion of G was equal to proportion of C. So this is actually highlights complementary base pairing, that A and T is complementary to one another, and G and C are complementary to one another. Name the bases ATGC. So AT is adenine, thymine, guanine, and cytosine. You think that was a pretty straightforward question. The next part actually deals with suggest and explain how the Shergas findings help Watson Crick work out the structure of DNA. So from Shergas findings, what was evident? It was evident that the proportion of A and T and G are stayed equal. This helped Watson Crick to link this with the idea of complementary base pairing. And when they linked it with complementary base pairing, DNA was then found to be having double helix. And whenever hydrogen bonds do exist between complementary bases that stabilize the DNA molecule, so they were able to consider the previous findings together with what they were adding to the new science at that time. The next is they have mentioned this Phoebus Levine isolated nucleotides of DNA and identified carbohydrate component of each nucleotide. So it's a DNA and the nucleotide having the DNA in the DNA, and the carbohydrate which is present, name the carbohydrate. Of the carbohydrate is a pentose sugar, it is a deoxyribose. If you mention pentose here, that answer will be wrong. So you need to make sure it's a deoxyribose because it's a DNA. Before the discovery of the structure of DNA as a molecule of inheritance, scientists thought that proteins were the most likely to be molecules that carried information. Suggest how the structure of protein made scientists think that they were the molecules that carried information. You need to suggest. So we know that same and similar proteins in all organisms do exist of same species. There are slight variations in the structure of the proteins due to different R groups and proteins. As amino acids are present in the sequence. So these may have contributed to for the scientists to think that probably proteins are the compound or the molecules of inheritance. But this was way before the DNA was realized at being the actual molecule for DNA for inheritance. Let's move to the next question. Okay, so this is May June 2019, variant 21. This question is about the DNA. The nucleus is known as the nuclear DNA. In cells of grasshopper we know as an animal, 20% of nucleotides in the nucleus contain thymine. So if 20% of 20% nucleotides are thymine, that means thymine is complementary to adenine. So adenine should be 20 as well. So 20 plus 20 is 40. It means that adenine thymine together are 40. So if you subtract this from 100, that means 60% of nucleotides will be guanine and guanine and cytosine will be equal. So 60% half is 30. So it means that the guanine and cytosine will be 30% each. Calculate the percent of nucleotides in the nuclear DNA of this organism that contain guanine. Explain your answer in terms of structural DNA. So 30% nuclear DNA will have guanine. So what's this? Out of 200% nucleotide, 20% are thymine. Since it bonds with 20% adenine, remaining GC pairs will be 60%. Half of 60% gives a 30%, which means 30% guanine. Therefore, 20% T and 20% A, 30% G and 30% C is equals to 100%. State another location other than the nucleus where the DNA will be present in this organism. Since this organism is an animal, it's a grasshopper, so we'll be mentioning mitochondria here because mitochondria is having carries small circular DNA. We can't mention chloroplast here. Figure 6.1 shows a diagram of a tRNA molecule. The region labeled in tRNA molecules is a part shown to you. So this triplet is anticodon. So tRNA is having anticodon. This region will be having complementary base pairing on the same strand of on the same strand. That's why it folds up. So if it's having C, C, A, G, the opposite of these will be C complementary to G. The next C complementary to G again. A will be complementary to U and not to 2 because it's an RNA. It's an RNA-RNA interaction. Both strands are RNA. So adenine cannot pair up with thymine. So it must be U, and then G will pair up with C. So complete figure 2.1, 6.1, we have completed. State the name of the region to explain the role of Q in translation. So it is an anticodon. We know that anticodon, this triplet of bases is complementary to a specific codon on mRNA. The tRNA or anticodon is always complementary to an mRNA codon. The complementary codon and anticodon attached during translation via hydrogen bonding of mRNA on the ribosome. State the function of region P. The region P, where is the region P? This region P is the one where you have the amino acid attached. So function of region P is to hold the amino acid and deliver it to the ribosome during translation of mRNA to form polypeptide. So I think that's how we cover tRNA here in this question. Okay, so the May June 2019 variant 23, they gave a question with this diagram, figure 1.1, draw a circle around monomer of a DNA. So monomer of DNA must have a phosphate group, it must have a pentose sugar, and it must have a nitrogenous base. So all three of components of a nucleotide must be there. So we will draw a monomer, the circle around these three. Okay, so name two bases forming the base pair at X. Since the base pair at X is having three hydrogen bonds in between, so this definitely means one of them will be guanine, the other will be cytosine, or this could be cytosine, this could be guanine, because they both are shown as a single square. So that's a guanine cytosine pair. You will mention three hydrogen bonds as reason. The stream 1 to 5 described events that occur during DNA replication. So 3 is the first that's happening. You need to state how that will form. So after 3, which is this is the DNA hydrogen bonds, they break. After that, there are two strands of the double helix. They separate out. Once they separate out, the hydrogen bonds they form, and when hydrogen bonds form, DNA polymerase they start to make their phosphodiester bond, and then DNA double helix forms. So this is the point which students get confused. What event occur first? So complementary base pairing by hydrogen bonding always occur first, followed by phosphodiester bond being formed by the enzyme DNA ligase actually. So not any polymerase, but although since the old syllabus used to follow DNA polymerase, so we don't really mention DNA polymerase anymore. The phosphodiester bond is formed by DNA ligase. According to the details that we are following from the new syllabus, which is from 2022 to 2024 exams. Okay, the telomere is a region found at the end of the chromosome after the function of telomeres. So telomeres are present at the end of DNA molecule. They prevent the loss of genes during DNA replication. They also prevent the joining of the two linear DNA molecules together. So that's the function of telomeres. Okay, so this is May June 20, variant 21. This is, you can see the transition protein eukaryotic cells shown to you. This structure is a ribosome, and the ribosome is going through an mRNA. It's reading the mRNA. These are the codons which it is reading. There are 4 codons which it's holding right now, and 2 codons can be read at once. 4 codons, even if they are being held, they can't be all read together. Only 2 codons can be read at one time. So you can see this codon having AAA will have an anticodon triple U. The codon UCG is having a complementary anticodon AGC, and they all have their own amino acids. So this is a peptide bond. The protein is actually forming here, and this is the next amino acid about to join. The bond between this will be a peptide bond, and it will be catalyzed by the enzyme peptidyl transferase. You can recall that this enzyme helps in formation of the bond, and this peptidyl transferase is present in the ribosome. So GUC is the next codon, and J codon, J is the tRNA end, and this is an anticodon. The sequence on anticodon should be GUC complementary structure. So G complementary will be C. So it will be C here. For U it will be A, and for C it will be G again. So it will be a CAG anticodon against a GUC mRNA codon. And this will bring the amino acid valine in place. So name the purine bases shown in figure 6.1. Purine bases are adenine and guanine, and that's a fact. You can just recall adenine and guanine. You need to learn that purine bases are always adenine and guanine. State the name given to the group of 3 bases found at J, so that this is the anticodon. Identify the 3 bases at J. We have identified these. State how the 3 bases at J and tRNA interact with the base on mRNA. They interact by hydrogen bonds. Let's take another example of question from DNA now. Okay, so in this question, this is from May 2017, variant 21. The DNA molecule is shown to you. You can see the 2 strands of DNA. One is running in this direction, the other is running opposite direction. This is the anti-parallel structure. These are the 2 hydrogen bonds, which means they are holding the base pair AT, and these are 3 hydrogen bonds. They are definitely holding guanine and cytosine together. So we know that adenine is having 2 rings. So this is a purine base, and similarly this is a purine base as well. So this is adenine, and this must be guanine. So that's how we actually identify. This is cytosine already mentioned. So coming back. Use figure 4.1 to explain how the structure of mRNA differs from DNA. You can mention size. You can say that mRNAs are shorter in size or smaller in size compared to DNA. There is 1 strand in mRNA. Thymine and uracil are present in DNA and RNA respectively. mRNA respectively. Double helical structure for DNA, whereas mRNA is looped. It can be looped, but it's never helical. So yeah, ribose sugar, pentose sugar is ribose and not deoxyribose. Complementary base pairing between strand from 5' to 3', and 3' to 5' in mRNA. The complementary base pairing along the same strand. So there are no two anti-parallel strands there for complementary base pairing. A similar question came here in Feb March 2022, and this question deals with the DNA molecule. Part of DNA molecule shown to you. Again, you can see this is a phosphate group, this is a pentose sugar, and this is nitrogen base adenine. So adenine will base pair with thymine, and two hydrogen bonds suggest that this is an AT base pair because AT is having two hydrogen bonds. Similarly, if it's a GC pair, so GC pair will always have three hydrogen bonds. They are saying identify structure E and F. Structure E and F structure E will be thymine because it's complementary to adenine, and structure F will be cytosine because complementary to guanine. Be very careful if it's a DNA molecule shown, the complementary base pairing will be in this way. If it's a DNA together with mRNA, then the complementary base pairing will be thymine cannot be there. Draw a circle around one of the nucleotides. So as I mentioned earlier as well, the nucleotide must have all these three structures together. So we will draw a circle around that. State the name given to the covalent bond that link the two nucleotides together, so that the name of that bond is phosphodiester bond. I think that's not that tricky. Figure 6.2 shows RNA basis sequence of a short primary transcript. Complete figure 6.2 by writing DNA basis sequence of template strand used to form the primary transcript. So the DNA basis of the primary transcript is to be completed, and the mRNA transcript is given to you. So for GGU, the complementary base pairing would be C, C, and A, for because this is DNA. So DNA cannot have uracil. So for GCU, it will be C against it will be C, and then C, and then G, and then it will be this is G. This is G here, and this will be U. And if you just go forward, if this is AAU, so this will be TTA. If this is CUA, this will be GAT. So that's how we'll just complete this complementary base pairing. Now in eukaryotic cell, primary transcript is modified to mRNA. Now this is according to the new syllabus. They have given this question from the new syllabus. So this question is about how the primary transcript is actually not the mature mRNA, how it changes. So they are asking you how how it changes. So what changes are there? So the primary transcript is having non-coding sequences too, and the non-coding sequences are called introns. The introns are then removed, they are cut and removed, and when they are removed, the coding sequences, which are called exons, they are then joined together to form final mRNA. So you will just make sure that you describe that, give a description of this and give reason of what changes are there, how the primary transcript changes to a mature mRNA. The mRNA strand is then translated at the ribosome to form a polypeptide. Describe how the process of translation results in the formation of a polypeptide. So yes, codon mRNA, anticodon tRNA, complementary base pairing, a mass is being delivered to the ribosome, peptide bond forming, peptidyl transferase enzyme. AUG is the first codon for every protein being synthesized. It codes for the amino acid methionine. I have described this in one of my videos in the section of my AS playlist, in which I am describing the nucleic acid and protein synthesis videos, which is chapter 6. So you can go and you can just try to follow why methionine is the first amino acid in every protein which is being synthesized. Although the final protein may not have methionine, it may get chopped off from the mature protein when the protein becomes functional. But yes, the first codon is always AUG, and it's always the amino acid methionine. Transition stops at the stop codon. mRNA is read in 5' to 3' direction. So these are the point that you can state here. I hope that this was helpful for you. That's it from my side. Thank you so much.